Everyone, and welcome to MycoTalks. It's hosted by the MRC Centre for Medical Mycology at the University of Exeter. My name is Alex Brand, and I'm one of the PIs at the MRC Centre. The co-chair today is Geraldine Butler from University College Dublin. Unusually today, both of our speakers are from the same university, the University of Wisconsin-Madison. The first is Professor David Andes from the Department of Medicine, Medical Mycology, Microbiology and Immunology. And our second speaker is Professor Nancy Keller from the Department of Medical Microbiology and Immunology. So MycoTalks are organized by the committee that's shown at the bottom of this slide. And we have valuable assistance running the event from Jules Bristow and Alberto Munoz from the MRC Centre staff. So MycoTalks are held at 1600 hours UK time on the last Thursday of every month, except for November and December, when they're brought forward a week due to Thanksgiving and Christmas. They were started by the MRC Centre in July 2020 during the pandemic, and we are now in our third season. Our participants span 51 countries, and MycoTalks have had 20,000 views on YouTube, so they're doing a good job at keeping the international medical mycology community informed about great science. The philosophy of MycoTalks is to promote diversity in our field in terms of career stage, gender and geographical location, as well as research field. The committee is always happy to receive speaker suggestions for our future programme. The format is two 30-minute talks on different aspects of medical mycology, followed by a 30-minute question and answer session at the end. To ask a question, please post it in the Q&A tab, which will be seen only by the moderators. It's important to state the name of the speaker you wish to address your question to. The chairs will alternate the questions between speakers, and they may choose to amalgamate some questions if they cover a similar topic. However, if we run out of time, all questions are posted to the speakers on the Q&A will be sent to them after the event, so they will see them. So the upcoming talks are listed. Oh, my thing isn't working. Right, the upcoming talks are listed on our website, and you must register for each session. Please note that the time of the talks is 1600 hours UK time. So in the spring and the autumn, please check how this might be affected by daylight saving status in your home country. And we've now launched MycoClinics as a series of online talks, specifically with clinical focus. And again, you can find the schedule for these on the MRC Centre website. And I also want to draw your attention to the Medical Mycology Trainee Seminar Series, which is held on a monthly basis and is a great opportunity for early career scientists to give a talk. So now it's my great pleasure to hand over to our first speaker. So David Andes is an MD and a professor in the departments of medicine, medical microbiology and immunology at the University of Wisconsin. He's also director of the Wisconsin Antimicrobial Drug Discovery and Development NIH Center of Excellence. The focus of David's research is antifungal drug resistance, and his studies span from the bench to the clinic. His work encompasses all aspects of this topic, from its molecular basis, its evolution, drug target identification and development, and strategies to combat the development of drug resistance in patients. Today, he's going to tell us about his work on drug resistance within the matrix of biofilms. So over to you, David.
Thanks very much for the kind introduction and the opportunity to share some of our work. Are you seeing the presenter mode?
Yes, that's perfect.
Great. Let's see if I can get a pointer here. Great. Well, again, I really appreciate the opportunity to share some of our work. I'm working with fungal biofilms, especially Candida albicans biofilm communities. These are quite common in our patients. In my practice, we're particularly interested in these biofilm communities that form on medical device substrates, particularly in our sickest of sick patients in the intensive care unit where vascular catheter infections are quite common. These communities persist in the face of antifungal concentrations that exceed the planktonic minimum inhibitory concentration several fold. And this has an extremely negative impact on our patients, where despite antifungal therapy, unless these vascular catheter biofilms are able to be physically removed, the chance of dying from this infection nearly doubles. Amongst the tools that Candida has to resist the effects of antifungals in these communities, our focus has been primarily on the extracellular matrix that these organisms produce and the community becomes encased in. And we're particularly interested in this phenomena whereby the extracellular matrix protects the community from the antifungals via a process we call sequestration, which is illustrated in this experiment where intact Candida biofilms are exposed to radiolabeled antifungal, and then we track where this radiolabel goes within the community. And the short answer is most of it is retained in the extracellular matrix and is unable to reach its drug target and impact the viability of organisms in the community. Our initial studies to understand this phenomena began with studies looking at the composition of this extracellular matrix. And as is observed with most microbial biofilms, the extracellular matrix is polymeric, it's quite complex. Our initial observation as it relates to the extracellular matrix composition identified a polysaccharide component, which was beta-1,3-glucan. Needless to say, we were disappointed to find that all of this work we had been doing on beta-1,3-glucan, you know, in that it was really a minor polysaccharide component. There are 3 polysaccharides that we find in the matrix: a branched mannan, which is the predominant material, followed by a beta-1,6-glucan and a small amount of beta-1,3-glucan. In an attempt to discern the role that this material might be playing, we utilized a candidate gene approach, looking at the putative function of based upon in Candida for either production or modification of these polysaccharide structures and the branching that they that had been identified. And we identified almost 40 mutants that we were able to create and screen these for biofilm phenotypes. And here's an example of one of these mutants that had a phenotype that was of interest to us. This is a homozygous deletion mutant in BIG1, which is relevant for production of beta-1,6-glucan. And we find that unlike wild type isolates that resist the effect of antifungals, in this case fluconazole, the homozygous deletion mutant was quite susceptible, and we were able to effectively reduce the burden almost completely in this mutant with drug concentrations not that much higher than the planktonic MIC, and that this phenomena was reversed back towards wild type with replacement of a wild type allele. Interestingly, this mutant also was able to, the extracellular matrix was less able to sequester the tritiated antifungal, and we speculate this is the reason why these cells are more susceptible in the biofilm state. And we actually found 10 of the 38 deletion mutants that that phenocopied each other in that they were hypersusceptible to the antifungal when grown in the biofilm, unlike the wild type. And a subset of these were in Manan modification mutants, mutants in beta-1,6 glucan production, and in FKS1, which encodes beta-1,3 glucan synthase. Because again of our interest in the matrix and the connection between drug susceptibility or drug resistance in the matrix, we then screened these mutants for extracellular matrix. As you can see here visually, the reference biofilms are encased in this fluffy extracellular matrix, while these 10 mutants do not produce visibly apparent matrix based upon scanning EM. We call these naked biofilms. So qualitatively, you can see each of these produces less extracellular matrix. When we look at these mutants biochemically, some of the observations we made were intuitive. So this is again data with the big 1 mutant, and we're looking here at the quantity of the 3 extracellular matrix polysaccharides, the Manan component, the 1,6 component, and the 1,3 component. And what was intuitive is that in this mutant in production of beta-1,6 glucan, that we would see less beta-1,6 glucan in the extracellular matrix. But what was surprising to us is not only did we see less 1,6 glucan, we saw less 1,3, and we saw less Manan, which was surprising to us because these production and delivery pathways are supposed to be distinct. And so this led us to wonder if these 3 polysaccharide materials are physically interacting in the extracellular matrix, such that if you lose one, you lose all 3. And in fact, that's what we found with each of these matrix mutants, regardless of the putative production or modification pathway that was involved. When you lost one, you lost all 3. We further explored the possibility of the interaction of these matrix polysaccharides in a combination or co-infection experiment, where we grew 2 strains together, one a mutant in one pathway and another a mutant in another pathway, and we did all combinations of this, remembering that when they're grown alone, they were naked biofilms and they were drug susceptible. But when you grew them together, the combination of these different mutants appeared much more similar to the reference biofilm. And this is, I'm just showing you what these look like visually. I'll tell you that when you look biochemically at the quantity of the different matrix components, when you grow them together, they are restored to wild type levels, and the biofilms are rendered drug resistant, again, similar to the wild type strain. Again, suggesting to us that they interact, and that also that they are likely assembled after they, or at least possibly assembled after they leave the Candida cell. But how this material is packaged and how it leaves and is delivered to the extracellular matrix remained really an enigma until we began to study the source and role that other extracellular matrix materials play. And we began study on the lipid component. And because of really emerging work in the mycology and other microbe field demonstrating the role of extracellular vesicles, we wondered whether or not a component of this lipid extracellular matrix might be associated with extracellular vesicles. And in fact, when we looked back at our early scanning electron micrographs using high magnification, we did find these spherical structures that decorated the surface of the Candida in the biofilm. And in fact, on the bottom left here, we can even see spheres in and adjacent to the extracellular matrix. So we isolated these spherical structures and found that they did appear similar to extracellular vesicles as demonstrated in this cryo-EM image, in this case, structured just under 100 nanometers. So we began to characterize these extracellular vesicles, comparing the extracellular vesicles that were produced and secreted in when the cells were growing in the biofilm state to when they were growing planktonically. And what you observe when you isolate extracellular vesicles from planktonic Candida growth is you see two populations of cells, a smaller population that's under 100 nanometers and then a larger population. When you grow these same species in the biofilm state, you see really a predominance of this smaller population of extracellular vesicles that are in the size range you would see with exosomes. Again, we also began to look at the cargo of these extracellular vesicles grown in these distinct environments and found that there were similarities and differences. So not only a difference in size, but some similarities and differences in cargo composition. Because of our interest in the extracellular matrix and in understanding how these materials were delivered, we then compared the cargo of these extracellular vesicles to what we find in the extracellular matrix. First, looking at the proteome, where again you see similarities and differences, but we find 45% of the matrix proteome is also found in the extracellular vesicles produced in the biofilm state, leading us to wonder if perhaps this is how they're delivered. We similarly looked at the glycome here, looking at just the H1 NMR of the Manan structure, where we see similarities, almost identical glycome components in the extracellular vesicles and the extracellular matrix. Again, leading us to wonder if both of these components are carried in the extracellular vesicles. To try and explore this further, we turned to the escort system. Again, because of the small size of the extracellular vesicles that we were finding and the importance of the escort pathway in other model systems for production of exosomes. So we created homozygous deletion mutants in each of the 21 escort pathway orthologs, and we screened them first for extracellular vesicle production, and 16 of them, excuse me, produced lower quantities of extracellular vesicles when grown in the biofilm state. And so we utilized these mutants to try and discern the role that these extracellular vesicles might be playing in the biofilm state. First, screening with imaging for the presence or absence or reduction, perhaps, in the extracellular matrix. And you can see here again, the wild type reference strain on the left, decorated, encased in this extracellular matrix, But this mutant in the escort pathway encoded by SNF7 is again one of these naked biofilms. And amongst the 16 escort mutants that had a reduced quantity of extracellular vesicles, we found 7 of them that had matrix defects. Again, not just visually, but biochemically, with almost complete loss for some of these in these mannan glucan components that we've linked to this drug protection phenotype. Again, in that vein, we then screened these escort mutants for drug resistance to fluconazole. And as we found with the matrix mannan and glucan mutants, we found these escort mutants were also quite susceptible to antifungal compared to wild type. And again, this was reversed with complementation. And each of the 7 escort mutants that had matrix defects, had vesicle and matrix defects, also were now rendered susceptible to antifungals despite growing in this biofilm community. So we wondered if this phenomena we were observing was due to the cargo of these extracellular vesicles, or whether or not this was just a pleiotropic effect of a mutant in the escort pathway. And to explore that further, we isolated extracellular and purified extracellular vesicles from wild type cells, and we amended these mutants with these wild type extracellular vesicles, and then again looked at these biofilm phenotypes. So again, here is data with that mutant SNF7 that you can see is quite susceptible to fluconazole when growing in the biofilms, but that is almost entirely reversed to the wild type state when we add back in wild type extracellular vesicles, suggesting that the vesicle cargo is functional and is responsible for this phenotype. And again, this is what we observed largely for each of the escort mutants. When we added back in wild type extracellular vesicles, you return the biofilm community to the drug resistant state. We did the same thing looking at our matrix assays. And so here you see visually the HSE1 mutant on the top is close to a naked biofilm, but when you add back in wild type extracellular vesicles, you can see the community begins to be decorated again with the extracellular matrix. And when you look at that biochemically, you see restoration of wild type and even above wild type levels of this mannan glucan complex. So our more recent studies began to try and understand the role that individual cargo might be playing in this phenomena. And again, turn to the escort pathway, where we found that not only is the escort pathway responsible for the quantity or production of extracellular vesicles, but also for packaging of cargo components, where we see quite a bit of differences in the protein cargo amongst the extracellular vesicles. There are a large number of proteins in these extracellular vesicles, and a colleague used machine learning to identify patterns and narrow down this potential list of protein cargo for us to explore. Narrowed it from over 1,000 to down to 63. And we were able to create deletion mutants for each of the 63, and then screen these cargo mutants for these biofilm phenotypes of interest. Now, when you look at the 63, we were pleased to see some of our friends that we had already identified as relevant for production of some extracellular matrix components, and others had been identified by other groups as important for the biofilm lifestyle. So we felt we were perhaps on the right track. And I'll show you some of the some of the data from this screen of 63. And in short, we found actually 17 of these cargo mutants had a biofilm phenotype, so a pretty high hit rate with this machine learning approach. And so here's data with one of these mutants. So this is a homozygous deletion mutant in PRA1, important for zinc homeostasis, that is hypersusceptible to the antifungal fluconazole. Again, reversed when we return a wild type allele. This mutant also has a matrix defect, as illustrated here. This is the polysaccharide component of the extracellular matrix. And when we add in wild type extracellular vesicles, we're able to begin to return these phenotypes back towards the wild type state, again, dependent upon the concentration of wild type extracellular vesicles that you add back in. And as I mentioned, we found 17 of these cargo mutants that exhibited these biofilm phenotypes, some of which we had already identified previously and were intuitive in that they were their function, their putative function is in either production or modification of a matrix component. Others were much less intuitive, at least as far as their putative function, and that's an area that we're continuing to explore now as far as the specific mechanisms that these proteins are playing in this part of the biofilm lifestyle. So I want to shift gears in the last few minutes and talk about sort of the serendipitous intersection of the work that I've just presented with our work in natural product antimicrobial drug discovery, the basis of which is utilization of microbial symbionts of insects and marine animals as a source of safe and effective natural products. And we actually find quite a few antifungals with this platform. And one of these antifungals we found we named turbinmycin. And it's a molecule that is produced by a component of the sea squirt microbiome, in this case, a Micromonospora species. And you can see there the structure of this antifungal and was named turbinmycin by the postdoc who discovered the structure based upon the species of sea squirt. And we were quite excited to find that this antifungal has activity against emerging drug-resistant pathogens, including those that exhibit resistance to all of our clinically available antifungals, suggesting, at least based upon this efficacy phenotype, that perhaps the mode of action of turbinmycin was unique, or at least different than the existing antifungals that are clinically available. And in data I'll now show, we showed that this is safe for animals and is effective in animal infection models of invasive candidiasis and aspergillosis. But what I want to share is our interest in the mode of action of turbinmycin and how it relates to, again, serendipitously to the work that we've been doing with the biofilms. And so using chemical genomics, in this case, a DAMP library in Saccharomyces, we find that the most susceptible mutants in this library were in the vesicle trafficking pathway. And the most susceptible was encoded by SEC14, which is a phosphatidyl and acetyl phosphatidylcholine transfer protein. And when we look more closely at the impact at the cellular level of turbinmycin, in this case, looking at Saccharomyces that have a SNCC1, a snare protein, GFP labeled. So what happens normally to SNCC1 is during growth, you see transfer of this protein from the Golgi to the plasma membrane of the bud. And that's what you see in controls on the left. But in the presence of turbinmycin in a dose-dependent manner, we see all of this GFP retained in the Golgi, again, consistent with interruption of the vesicle trafficking pathway. And again, this was interesting to us because we wondered whether or not turbinmycin might be effective or do the same thing in biofilm cells. And so, in fact, that's what we found. So this is data with Candida albicans, and this has been recapitulated with other Candida species and Aspergillus as well, is that when you expose the biofilm community to turbinmycin, you really shut off the production and delivery of the extracellular vesicles. And we looked more closely at the impact of turbinmycin on these biofilms and found just really low concentrations of turbinmycin, as you can see on the right-hand scanning electron micrograph, really reduces, almost completely eliminates the production of the extracellular matrix, again, at very, very low concentrations compared to wild type cells. And when we look in animal models of these device biofilm infections, again, in this case, a central venous catheter that we place in the external jugular vein of rats, we can see on the top right, that's what the intraluminal surface of these catheters looks like in control animals, just chocked full of biofilm. But if we expose these catheters to turbinmycin, we see really elimination not only of the extracellular matrix, but if we use a high enough concentration to treat these animals, we're able to completely eliminate the biofilm community. So we're quite excited about this observation, both as a tool to understand the biology that we're interested in these biofilms, but also potentially as an antifungal. And I'll just stop by summarizing. I think we've shown that the Candida biofilm matrix is an important mechanism for protection of these fungal communities, that this material is polymeric and includes a mannane-glucan complex that is associated with this drug protection phenomena. That this material and other components of the extracellular matrix is assembled in the extracellular space, delivered by extracellular vesicles, we think mostly exosomes, and that this is a drug targetable process, as we found with the antifungal turbinmycin. And so I'm going to stop there by thanking the folks who've done this work and our collaborators in funding, especially our collaborator, Erin Mitchell, who we enjoy working with greatly. And I will stop there.
Thanks, David. That was fascinating. So just to remind people that we will take the talks take the questions for both speakers after the second speaker. So please enter all your questions in the Q&A and we will deal with them later. So our second speaker, staying at the University of Wisconsin, is Nancy Keller, who's the professor of medical microbiology and immunology and bacteriology. And so Nancy's lab has a long history in studying Aspergillus and other filamentous fungi, and especially the function and regulation of secondary metabolism gene clusters. But today she's going to present her work on developing a computational network to explore Aspergillus fumigatus signaling and molecules directing hyphal development, host interactions, and interactions with the microbiome. And the network, which is called GRASP for gene regulation of Aspergillus fumigatus. And she's going to show us how that was used to recapitulate known pathways and also to discover new roles. So it's likely to be a very important tool for many in the Aspergillus community. So over to you, Nancy, and we're looking forward to hearing from you.
Thank you. Let me get this. I'm sharing, and Is this in the proper format?
Yes, it is.
Okay, let me get my, okay, well, thank you so much. I'm really honored and appreciate you asking me to present here today. And I'm just going to go with it. So this project I'm going to present today is a totally new project in the lab, and it's a collaborative project with the Shumita Roy Lab and the Jean-Michel Ane Lab. The work I'm going to show you today is largely unpublished, but we should be submitting this before the year's end. So we thought this would be a great little preview for everyone to get some feedback from you. So let me just start. I'm going to minimize my, okay, unfortunately, let me just 1 second here to minimize something so I can actually see what I'm doing. Okay, so this lovely creature you see in front of you is Aspergillus fumigatus. It's a filamentous fungus, always filamentous, unlike what Dave was presenting with Candida, though of course Candida is filamentous too, but this is obligate filament. And our lab, for as long as I've been working, has focused on natural products. Natural products, there's a lot of meanings for natural products, and there's certain ones that we're most interested in. We're interested in those natural products, which are actually developmental signals to the fungus. So Aspergillus and other fungi will produce metabolites, which are secreted and then perceived on the outside of fungus by some receptor to direct the fungus into the right growth form at that particular environmental niche. For example, you might find signals that say germinate, or in filamentous fungi to branch, or when to not to branch, or when to sporulate. So that's one of my great interests that we'll be talking about today. And of course, my lab has long been looking at microbial secondary metabolites. Aspergillus and many filamentous fungi have these fantastic biosynthetic gene clusters that encode and make metabolites. And these metabolites, actually, Dave brought them up a little bit, though he was talking about one from a bacterium. These metabolites, for the large part, are really weapons of the organism that makes them. For example, in Aspergillus, it makes a whole slew of metabolites, which majority of them are antifungal or antibacterial. However, some of these are also harmful to the host. In the case of Aspergillus, you really don't want to have these metabolites being produced as the fungus grows in you. Some fungal metabolites are quite helpful, but we're not going to be talking about the helpful ones today. So keeping this in mind, this program, GRASP, that I'll be talking about has been put together with the thought that it can actually help us answer biological questions in Aspergillus fumigatus. Let's start with the developmental signals. So our lab, in collaboration with the Ane lab, with which I'll go into some detail, has identified two lipid-based molecules that actually regulate branching in Aspergillus. You can appreciate that when a spore germinates, you can't just have one hyphae that keeps extending onwards and onwards and onwards. That's not going to help the organism. Likewise, if it starts branching too much, the colony remains very small and cannot expand. So the filamentous fungi have had to develop a program that balances and tells them to branch at the right amount, lateral branches. What we found and published about 2 years ago is that the fungus makes a series of oxygenated fatty acids called oxylipins, and we found 2 of them that actually induce hyperbranching. And then the fungus also produces a lipochitooligosaccharide, which does the opposite. It induces hypobranching. Let me give you a couple examples. So this is adapted from our paper that was published a few years ago. PPOA is an oxygenase that will take fatty acids and produce oxylipins. It produces two primary oxylipins, 5,8-dihode and 8-dihode. As you can see in these bar graphs, as you add increasing amount of dihode and to some degree 5,8-hode, there's an increase in the branches of the fungus. And in fact, we usually use two common strains of Aspergillus that labs all over the world work with, AF293 and CA10. They have differential virulence. I'll point out now that Dr. Bromley is going to be talking in January. He's in charge of making the complete knockout library from this strain. But anyways, just going back here, you can see by adding these oxylipins, you triple or double the branching of both strains. And what you're seeing here is just a nice little example of the visuals we have of seeing increased branching. We became very interested in this with the thought that branching is not only important in the environment as a saprophytic fungus, but also as a pathogen. So we performed RNA-seq, treating the fungus with a 5,8-dihode, and we found that this compound actually both induces and represses a lot of genes, as you'd expect. In fact, so many, we had a little hard time knowing where to start. Well, luckily, at the time we did this study, Dr. Bromley's group had already deleted all of the transcription factors in CEA10. So we collaborated with him, and we looked at the 35 transcription factors that were differentially regulated by 5,8-dihode. And we found one that was the gem. It's a B-zip transcription factor called ZFPA. If you look here in this calcifluor stained slide, you can see in the wild type, this is the carrier, ethanol. Here we see a normal Aspergillus. When you add the 5,8-dihode, you can see the increased in branching, and you'll probably note the increased staining with calcifluor white, which is a chitin binding dye. What we find is if you overexpress ZFPA, the fungus is locked in a hyperbranching phenotype, and if you delete it, it's locked in a hypobranching phenotype. And in either case, whether you delete or overexpress, it's totally non-responsive to dihode and 8-hode. So this became very critical to us. We found this transcription factor, the first to our knowledge, that is in charge of regulating the balance of branching in Aspergillus. Now, as a little teaser, because I'm asked this all the time, let me just summarize here. So we were able to show that the oxylipins actually go through some signal transduction pathway, what's in this blue arrow space, we don't know, to induce this transcription factor, ZFPA, which is involved in the regulation of branching. We have looked at this fungus now, the deletion and overexpression, and I'm just going to give you one teaser little slide, as this actually is another project, but you might be interested. It is a virulence factor. This is a figure from, I almost called you, I don't know if Taylor's on, my student, I called you Dr. Schoen, soon to be. Taylor Schoen is a joint PhD student with Anna Huttenlocker, who's a zebrafish immunologist, and Dante Carlisle is a PhD student in my lab, so this is a joint work with them. But what you can see here is that if you just look at the solid lines, you can see that in a wild type fish, RAC2 wild type, it's wild type fish, the deletion of ZFPA gives a decrease in virulence. The overexpression ZFPA was largely the same as the wild type. through various other studies that I'm not going to show right now, Taylor got the hint that maybe this was due to neutrophil activity. So she then took a fish line that the RAC2 D57N, which is deficient in neutrophil activity. And you can see when we look at that fish line that there's no longer a virulence defect in ZFPA. So there's a lot of interesting interactions with ZFPA, the loss and overexpression in neutrophils, which largely seems to be associated with cell wall. But as I say, that's another story. I just am showing this to show you we're very interested to know more on how ZFPA works. Now let's switch for a second to our other signaling molecule, the lipocyto-oligosaccharide, which from now on I'll call an LCO. This particular project is a collaboration with Jean-Michel Anne. Dr. Anne works primarily with fungi and bacteria that interact with plant roots. And so LCOs had been known for some time. They were compounds that were found to be produced by nitrogen-fixing bacteria and mycorrhizae, the root fungi. And the thought was that they were only produced by those organisms. But that's not the case at all. Another joint student of ours, Dr. Thomas Rush, actually found out a couple years ago that almost all fungi produce LCOs. And why would that be? Well, it's not easy to genetically work with the mycorrhizae fungi. So as we saw that Aspergillus fumigatus makes LCOs, we decided to work with that organism. And what Thomas found is that in contrast, oppositely to what we saw to the oxylipins, is when you add one of the LCOs, a sulfated LCO, you can see right here, you had hypobranching. So this comes back to what I was originally showing you, is that we have these two systems, oxylipins that induce branching, LCOs that inhibit branching. So our question now becomes, what are the genes and pathways? How do we start filling in the pathways that are illustrated by these blue arrows? Well, there's lots of things we could do. Some more RNA-seq, some proteomics, mutagenesis, go through all the deletion library. I can tell you that's a lot of work. And then that's we said, is there a way we can do something that will give us a better vision on the pathways and the environmental signals that actually set up this type of developmental phenomenon in the fungus? And that's where we turn to this computational program. And I really have to say that the algorithms and mathematics all come from Dr. Shamisha Roy's lab. I will not be able to answer any questions on algorithms, but this is the general flow of how this works. What we did is we took all available RNA data sets of Aspergillus fumigatus, and that included about 300 separate RNA-seq samples. And then we take that information, and we also take the information of DNA sequence motifs of transcription factors or other global regulators. It is then put through a program that Dr. Roy's lab has developed called Merlin. And this I will not go through. The paper is going to be submitted on this very shortly as well. But the main thing, the important thing for the viewers today is that the output gives us some very useful information on modules of genes that are co-regulated at least 80% of the time, and relationships with these genes. But let me just show you what we got out. So to see if this Merlin was useful, we wanted to gather the information. Here's the first output. Now, many of you have seen these very complex hairballs, and we recognize, oh, this is really hard to interpret. So what we then turn to is ask, well, what modules, each one of these little dots with a little dots connected to them are a module, where we would have what would be, you can think of it as a regulatory gene that seemed to be regulating a subset of other genes, our target genes. And what we found is that there were about 5 modules that regulated over 100 genes. And for example, one of the, not surprising, transcription factors that had a big impact on a lot of genes in Aspergillus was a mating type factor. We found 34 modules that regulated 20 to 100 and 125 that regulated less than 20. I actually sort of like these ones. It gives a little easier way to get going. You can see that of these, we got significant statistical data of regulation of about 3,000. Now the fungus has 10,000 genes, so we didn't get all of them yet, but this is a start. The beauty of this program is we can keep adding more and more RNA-seq and ChIP-seq, and that would be our goal. But we wanted to start somewhere. Now, we knew for this to be a program that I could easily understand, and I think a lot of ones, we needed some visuals. So this program actually will be working on the web, and hopefully you will see it and enjoy working with it yourself. So here's what you would see if you'd open this up on your computer. You can search by at least 4 parameters, but the most useful is a gene list. So you'll have this program and you say, I want to look at a gene, and then you put in your gene. For example, we decided to look at SRBA, the sterile regulatory protein. This is a protein that Rob Kramer's lab has really done a lot of work with, but other labs too. So this is what you do. You put in the gene of interest, and up pops up this beautiful display. And every single one of these dots is a gene here. And it's telling you that all of the genes you're seeing right now are expressed at least 80% of the time identically in the 300 RNA-seqs. The other piece of information is every time you see a red dot, it's a regulator, and most likely a transcription factor here. So SRBA. Now, what other information will you get from this? Well, if you scroll down on this, it's going to tell you the name of every single one of those blue dots. For example, you can see this first one right here is sterile reductase, which was a really good thing because we knew that SRBA is supposed to regulate that. And then you can we have this program that you can just click on that gene and it will take you right to fungal DB. Very useful to know more about that gene. But let's go back to SRBA. So here I've just taken the those modules, those genes, and showed you what some of them are. Well, when we looked at this, we were actually pretty ecstatic because for Merlin, for this grasp to be useful, we should be able to recapitulate what's already known. And sure enough, what we find, the genes that are known in the genes that came out all fit actually exactly on what the Kramer lab had published in 2014. We find that SRBA interacts with its other binding partner. It regulates the ergosterol pathway. It regulates the nitrate pathway and HIM13. What the beauty is now is what are these other dots? And maybe this is something that when it goes, we publish this, Rob's lab will like to look at. We wanted to check that there were other known pathways that were recapitulated, and these are just a few that we're showing you. CFA is a transcription factor that regulates known 3 zinc transporters, and here they are. HAPX is a transcription factor that regulates iron homeostasis in the organism, and these are all of the siderophore proteins. FAPR, actually, we identified in our lab as a transcription factor that regulates fumagillin and suritin biosynthetic genes, and there they are. This gave us confidence to then go with our own wet lab experiments. So let's go back to what I really was interested in. Can GRASP help identify pathways involved in oxylipin and LCO signaling. So let's look at the LCOs. We did an RNA-seq of treating the fungus with LCOs. We then took the genes in that RNA-seq and asked, do they overlap any of those modules in the GRASP program? And it was very clear that the green genes, which are regulated by LCOs, are all in the ATFA module. ATFA is a BZIP transcription factor that is known to regulate stress responses in all the fungi that has been looked at. So we asked, is ATFA involved in response to LCOs? The answer is yes. What Chris did then is, using his wild type control, he treated it with LCOs, and as always, you can see a decrease in branching. And then he looked at a deletion ATF strain and an overexpression ATF strain. And you can see in both cases, there's absolutely no response to LCOs. And in fact, who would have known this, because nobody looked at branching before, the deletion ATFA is a hyperbrancher, and the overexpression is a hypobrancher. Sort of reminds you a little bit of the ZFPA background. So we were very excited about this. And this has allowed Chris to start a visual idea of how LCOs signal hyphal branching. And I'll also say they signal biofilm and germination. So we know LCOs are somewhere in the outside, and they must be interacting with something in the cell wall and are transduced to ATFA. And I would say Chris has a few clues on how it's being transduced. But let me go on to another example. So I told you one thing I was interested in additional to the developmental signals are the natural products that this fungus uses as weapons. I'm going to throw out to you that Aspergillus fumigatus is probably the best characterized fungus in knowing exactly what its secondary metabolites are. This fungus has 8 chromosomes. You can see them here. Everywhere you see a blue band in the chromosome is where there's located one or more biosynthetic gene clusters. I could spend a lot of time talking about them, but I'll restrain myself. We and some other groups worldwide have been able to take these biosynthetic gene clusters and characterize the metabolite they make. And so all of these metabolites here are compounds that are produced by a specific biosynthetic gene cluster. Moreover, and importantly for anyone interested in this fungus, or I'd say filamentous fungi, we know what these do. Of these compounds circled in red, every single one of them has had some implication in virulence of Aspergillus and the development of aspergillosis. Sometimes it's minor, sometimes it's quite significant. I'm going to show you how GRASP has helped us understand the regulation of these compounds. And I'm going to focus on the most famous natural product from Aspergillus fumigatus called gliotoxin. Here you can see gliotoxin. It's a nice little small metabolite that you do not want to be near. So gliotoxin is very toxic to all eukaryotes. Remember, I told you that these compounds that fungi make are often antifungal or antibacterial. This one is antifungal. And in fact, when gliotoxin, the chemical, was found, which is about 1950, I think, it was thought, oh, maybe this can be a good antifungal. Well, it is true. It's a wonderful antifungal, but also kills people. So it was discarded. Now, the fungus, it's a fungus too. So how does it protect itself from its own gliotoxin? This is really a fascinating story. This is the gliotoxin biosynthetic gene cluster. If you look down here, our lab was the one that characterized GLZ. GLZ is a transcription factor that induces the expression of all these genes in this pathway, except for one called GLI-T. Well, GLI-T is one of the self-protective genes that Aspergillus fumigatus expresses. The pathway actually makes, pardon me, excuse me. So GLI-Tox, as I said, is the toxic metabolite, but it needs to be made by activity of GLI-T, and GLI-T, if it's not expressed, you don't get gliotoxin, but you get diethylgliotoxin, which is not toxic. And then the fungus also has another gene called GTMA, which will convert diethyl to bismethyl, which is not toxic. And that's how the fungus protects itself. Maybe pay attention to this name, GTMA and GLI-T. So even though we knew that GLI-Z turns on this pathway, with exception of GLI-T, we don't know what tells GLI-Z to be turned on. And that's the truth with all these natural products. So we went to GRASP. What I want to show you here is that you can put in more than one gene. So if I put in GLI-P or GLI-Z, any GLI gene, you get what's is called the gliotoxin module. All the genes, if you look at GRASP by module, they'll show you what genes are co-regulated and one color. But I was curious because we have this hypothesis that the fungus co-regulates a subset of these natural products for a 1-2 punch, basically synergism with these natural products. So I put in a gene from the fumigen pathway and the flumitremerogen pathway. And this is what happens. You can see that we've got 3 modules of these different toxins, but they look like they might be connected. If we switch on GRASP and hit regulator instead of module, the colors of the modules go away and we get the red regulator genes. And so what you can see here, if we just sort of look up at GLI-Z, is there's GLI-Z, it's a transcription factor. FAP-R, the one that we already know, it's a transcription factor. But we had all these other red dots. What are they? So for example, I'll show you what we did. So there was a transcription factor here, which is suggesting it regulates GLI-Z and the gliotoxin gene cluster. Never heard of it. Who could have even guessed? It's nowhere near where the gliotoxin pathway is. So we deleted that gene. And let me just give you a little, here's showing that module where we actually name all the genes. And you can see all the gliotoxin genes are here. And so when we deleted this gene, we looked at 2 of the products of the gliotoxin pathway, the toxic gliotoxin, the non-toxic bismethyl. And you can see there's about a deletion, 50-fold deletion. We are very excited about that. I am going to throw this one that we're working on now. We haven't deleted this one yet, but these are the 2 the 2 enzymes that protect the fungus. And to us, it's rather interesting that they seem to be regulated, of course, by the gene we deleted, but also they seem to have perhaps a transcription factor that's specific for the resistance genes. That we'll be looking at shortly. So I'm going to wrap up what I have shown you now and tell you what we're doing next. So using GRASP, Chris is very interested in filling up all of the question marks between the LCOs and the ATFA. He's found that GRASP has implicated I would say a kinase that he is looking at. Now, GRASP cannot answer everything. As I showed you in the very beginning, right now there's only 3,000 of the genes that we feel are that we can predict very strongly. We hope that everybody in the audience and the world that works with this fungus will keep adding RNA-seqs and we can put it in and enlarge that number. So there's some logic here, but one of the main goals is to actually completely understand how LCOs are inducing ATF leading to this phenotype. What else are we doing? near and dear to my heart. I think many of you know that I've been working with secondary metabolites for, I guess, my whole career. And it's easy enough to find a biosynthetic gene cluster and to understand how that gene cluster is regulated. But how and why the fungus is making them, not so easy. We're so excited to look at the potential of understanding synergism. Is the fungus actually, depending on the environment, co-producing one or more of these natural products to have an advantage in the environment it's in? And I think some of these regulators, we hope, we will be deleting and overexpressing all of them, is going to show us this. This might even give us some insight into antifungals that you could actually use in the clinic by showing us what chemistry together is effective for the fungus as it challenges other microbes. And of course, I don't want to forget ZFPA. I showed you in the very beginning that this is the transcription factor targeted by oxylipins. It's a virulence factor. How is it regulated? What other pathways is it involved in? And so we have quite a few candidates to look at that for that particular project too. So I want to just end by giving my acknowledgements here. This was for those of you who actually have fun for Halloween. This is what we dressed up in our lab, the Angry Birds for this Halloween. Just thought I would share that. But let's go to what's important here. Well, having fun is important too, but in my lab, whose work I showed you today was Dante Khalil, Chris, who's a Cariel, who's a joint student with Dr. Ane, Dr. Sensual Park, who did the chemistry with gliotoxin, and Taylor Schoen, who's a joint student with Anna Huttenlocker. Dr. Sushmita Roy, her lab is just incredible in developing these programs, and her PhD student, Spencer Hallberg, and her postdoc, Saptarshi Pine, were the ones who actually put together this fantastic program for us. And of course, as I said, we collaborate with Dr. Ane and Dr. Huttenlocker. And yeah, that's it. So thank you very much for your attention.
Thank you very much, Nancy and David. Fantastic talks. Okay, so we'll go straight into our Q&A session. And if I can remind everybody, please, to post your questions in the Q&A tab and make it clear to whom your question is addressed. Okay, so I'm going to kick off with some questions for David. Right, David, is the high degree of drug sequestration that you see in biofilms also seen with other drug classes, or is it somehow unique to fluconazole or at least the azoles?
Yeah, that's a great question. There is variability in the efficacy of the different drug classes against biofilms. And the antifungal class that is most effective at treating biofilm, at least in Candida communities, are the echinocandins. That being said, we do observe sequestration with polyenes, with flucytosine, and with the echinocandins as well. So it is a phenomena. The exact mechanism underlying that interaction, it's not, remains elusive and something we're working on, but it does appear to be a process that is important across drug classes.
Okay, right. And then there's a couple of questions on a similar theme here, so I'll merge them together. So biofilms are in vivo polymicrobial usually. And is it known how the extracellular matrix of Candida is affected by bacteria? And do the fungal EVs that you describe have any influence on other microbes, in particular uropathogenic E. coli in the bladder, that might be trying to establish on catheters?
Yeah, that's a great question. So, yeah, the interkingdom interactions of organisms in the biofilm state is fascinating and common. In a collaboration with Marianne Jabarisk, they showed that the extracellular matrix of Candida albicans actually protects Staph aureus from antibacterials. Interestingly, but that's not always the case. So with strep mutans, for example, in the mouth, when you see biofilms producing caries, it's the strep mutans extracellular matrix actually that sort of drives the biology of that interaction. So lots and lots of variation depending on the community members that you find there. We are interested in understanding the role of extracellular vesicles in these intra-kingdom interactions. We've begun some studies with Staph aureus. We have recently shown that across Candida species, the extracellular vesicles from the different mixed species components can and do interact and impact the biology.
Okay, so there's definitely interaction there. Okay, I'll hand over to Geraldine.
Okay, so Nancy, one question. What do you think the advantage of branching is? Is it to increase that absorption of nutrients, protection, or some other properties?
Okay, yeah, all of those actually. Really nice question. So when you branch, every time you branch, you get a new tip. And in filamentous fungi, the tip is where you both secrete metabolites and degradative enzymes and where you bring in compounds through the endocytic collar. So in fact, I will say in industry, they're always looking for hyperbranching mutants because you can get a lot more enzymes of whatever you're looking for. In general, you get more natural products. So you have that, the secretion and absorption, but also as you branch, some of the lateral branches fuse with an older branch that was a little further away, and it gives a stability to the fungus as well. You definitely want to branch. And in some cases, maybe hyperbranching is a good thing for the fungus.
Okay, thanks. So Alex, you want to go now?
Okay, right. David, so there's another set of questions around sort of regulation, really. So do you think that the extracellular vesicles play a role in initiating biofilm formation? And similar to that is it specifically linked to biofilm formation, and what could be the signal?
Yeah, that's a great question. We actually had a paper come out yesterday on this. So we've looked at the role of vesicles in adhesion, matrix production, and dispersion, and they do play a role in each of those phases of the biofilm lifestyle. Mechanistically, how in this case adhesion is impacted by the extracellular vesicles, we don't know. The organisms really start to ramp up as soon as they adhere. Within 20 minutes, the population of extracellular vesicles. So I showed you early on when they're growing planktonically, you see these two populations of extracellular vesicles, the exosomes and the larger. Within 20 minutes, that population, that bimodal population shifts entirely to exosomes. Within, so it's within minutes you start to see a shift in the vesicles that are being produced and their cargo.
Wow, right, okay. I'll go back to Geraldine.
Right, Nancy, so we have a couple of questions about the limitations. So what do you think the main limitations are of the database, and how accurate is it when you're studying something that's completely unknown?
Well, the limitations, as we just started this, it's hard to answer some of it, but right now the limitation, as I saying, is associated with lack of RNA-seq and ChIP-seq, because we only got 3,000 of those genes. So I think as we do more, get more data, in fact, I've got a couple RNA-seqs I want to put in right now, we will increase the number of genes that we can place into the network and actually have a more statistical confidence in the ones that we have put in. As we've only looked at a couple right now, 3, I showed you 2, but we've done 3 and they have been what we predicted. That's pretty good, but that's only 3 out of many. Ask me again in a few months, because we're going through all the ones that I think could be interested in secondary metabolites. Well, we really want to get this out, because I could see a lot of people, I know specifically what they're doing in their lab, that I think would like to look at some of these genes.
And so all of your motif data, that's all from ChIP-seq experiments?
No, some of it is just predicted. When we had a gene like CREA, for example, CREA is the carbon catabolite repressor transcription factor in all fungi, honestly. We can look, when we have a conserved protein like that, we actually looked at some of the yeast database and put in a motif that maybe wasn't quite clear in Aspergillus, but seems strong in yeast. So, you know, I'm sure we'll have things that won't work, but we're feeling, I don't know, I'm feeling really pretty good about it so far. But yeah, so some of those are from motifs that have been demonstrated in Aspergillus fumigatus. Of course, we had those Aspergillus nigilans, that fungus has helped us quite a bit. And then, you know, we're going out a little bit on a limb, but there's motif finding programs that help you quite a bit in this. This is all an experiment.
Okay, thanks. Yeah, Alex.
Okay. Another sort of merging of questions here, David. This is to do with interactions within the biofilm. So the biofilm matrix is generally hydrophilic, whereas EVs, the assumption might be that they're hydrophobic. So what mediates their interaction, and how do, I mean, if they carry cargo, how, you know, they're floating about, but unless they've got transmembrane domain with protein things sticking out from the outside with a target, what anchors them and releases their contents? And then on top of that, are they fusing with microvesicles? And do they need to do something like that to be effective?
Those are each great questions that we do not have the answer for. I can tell you that there are proteins that coat the surface of these in addition to intravesical cargo, and whether or not those play a role specifically. But we know we can sort of, with proteases, sort of shave off the surface proteins, and but interesting avenues to explore for sure.
Yeah. Have you got transmembrane proteins in there?
Yeah, for sure.
Yeah. Okay. Right. Okay. Geraldine.
Hey, Nancy, amazing talk. Do you know if A. fumigatus also makes lipid mediators belonging to the eicosanoid family? And second question, if oxylipids or any other lipid mediators derived from blood cells could affect the potential of fumigatus to develop and germinate in the blood?
Well, thank you for asking that. Yes, the fungus does make some eicosanoids, and we have a big program in our lab on looking at that. Actually, some of the human eicosanoids do affect the Aspergillus, and we actually are exploring. I have a postdoc giving a talk tomorrow in the lab on how there's crosstalk between these eicosanoids and eicosanoid-like metabolites that Aspergillus makes and the host makes, and the receptors on either side. So we think it's part of the virulence program of this organism.
Okay, it's another story.
Right, another story.
Right, David. One question here is, do the mutants you have that have defective biofilm matrix formation also have defects in other secreting other compounds like Candida lysine?
Yes. More, there's a paper submit, yeah, more to come. There are some, these vesicles carry a lot of cool things.
Okay, a guarded answer, I suggest, though. But okay, right, back to you, Geraldine.
Right. So rhamnolipids Pseudomonas affect branching. Is there any possibility they work through a similar pathway?
Again, thank you for asking that. We are looking at that too. We've got some, not in that particular protein, but we're very interested in Pseudomonas Aspergillus crosstalk at the lipid level. And so we've started to look at some of the lipid mutants we have in fumigatus, and then we have gotten the Pseudomonas oxylipin mutant. And I could say we already see there's a difference in biofilm formation of these different organisms. So there does seem to be crosstalk with the lipids of the microbes, which actually makes some sense, I guess.
Alex?
Okay. David, have you, outside the context of your biofilms, if you put these your mutants into a virulence model, are they avirulent? You're muted.
We've put the mutants into our in vivo biofilm models, but not in the more traditional mouse virulence models, but that would be interesting to do for sure.
Okay, right. Okay, Geraldine?
Nancy, do the hyperbrancher mutants also produce higher amounts of extracellular vesicles? So have you ever looked at that?
We got to get David to do that for us.
Yeah.
I suspect so. One thing I didn't tell you, because there's just so much in this time, is that biofilm formation is greatly impacted. The hyperproducers seem to have quite a bit more biofilm, and again, this will be coming out in this one zebrafish. The overexpression ZFPA, the hyperbranching, is much more resistant to antifungals, and the deletion is more sensitive. So, David, we got to go back to looking at some ECs again. EVs, pardon me.
Okay. Right, another question for David. Have you observed a different orientation of hyphae in the ultrastructure of your biofilm in the matrix mutants? For example, do hyphae grow parallel to the bottom of the well instead of forming a better 3D structure?
So the short answer is, yeah, I mean, when I think back, there are interesting intact biofilm morphology differences amongst mutants. We've not explored that systematically, but that would certainly be interesting to do.
Okay, right. Yeah,
Okay.
Geraldine.
So Nancy, are these oxylipins and LCOs produced by other species, so outside the filamentous fungi?
Oh, yeah. Candida doesn't make them. Saccharomyces doesn't make them. The dimorphs should make them. They've got the enzymes to make them, like histoplasma. No one's tested, but the filamentous Ascomycetes definitely make them. We're not sure about the Basidiomycetes right yet.
Okay, thanks.
David, have you any idea what nucleic acids are in the fungal EVs? I think there's an assumption there, but you'll know the answer.
Yes. There are nucleic acids in these EVs, which I guess isn't surprising at all. The role that they're playing is something we're interested in understanding.
Okay, right. Geraldine.
We have a long question from Paul Dyer. So really exciting to see the potential to identify so far unknown interactions. But regarding the RNA-seq training sets, how do you allow for the fact that under certain growth conditions you'll see interactions, but perhaps not under others? So are you using pooled data which might overlook some rare growth interactions, or can the algorithm still pick out interactions from rare RNA growth sets? I'm sorry to ask questions about algorithms.
Well, Paul, I'm going to refer you to Dr. Roy. You should send me an email and I'll give you her email, because most of those things, the string, I can't answer. I do know from talking with her student and postdoc that we're trying to allow for those rare interactions. But I think that will be helped if we can get some more RNA sets from different types of growth conditions and interactions. So, but I think, get hold of me and I'll introduce you to Dr. Roy for some details on the program.
Okay, so David, I have a question from my own experience with biofilm. So have you ever tried treating your biofilms with a specific, I mean, if you accept that you've got the chemistry of the cell wall, you've got a lot of polyphosphate, lots of negative charge, whatever, it's very hydrophilic. I just wondered if you tried pre-treating or the walls of the cells to see whether you get either a change in drug, in drug sequestration, really.
So yeah, that's not something we've done recently, but we have treated with different hydrolases, specific hydrolases, and shown that impacts the extracellular matrix and the biology of the extracellular matrix. So if that's what you mean, different monocytases and things like that. Is that what you mean?
Well, I know we tried using, sorry if this is turning into conversation, we tried using, if you can wash out, if you use EGTA or something like that, you can wash out a whole load of ions, or you could wash in a load of things that would bind lipids. I just wondered if you'd tried that. Yeah,
so we've done, I mean, relative to that specifically, we've tried different chelators, and that has a big impact on biofilm formation. Teasing out, since most of these chelators are many of them are non-specific, we've been stuck. We've been trying to do, add things back and see what is going on, but the short answer is, yeah, it has a big impact, and I think can be important clinically.
Yeah. Okay. Right. Geraldine.
Okay. Do the hyperbranching mutants have more septation than the wild type? Are they hyperseptate?
Yes, they're hyperseptate.
Okay.
Yeah.
Nice short answer, because we're running out of time.
Yeah. So I think we've come to the end of our questions. So I would really like to thank David and Nancy for fantastic talks, and also Geraldine for being the co-chair today. I also want to thank the staff at the MRC Centre behind the scenes who are actually in ultimate control of the cameras and things, thank goodness. I would like to announce the next MycoTalks is on 15th of December. And the speakers will be Professor Carol Kumamoto from Tufts and Professor Richard Bennett from Brown's University. So thank you very much for listening, everybody, and we hope to see you on the 15th of December. And thanks to our speakers again.
Thank you.
Take care. Bye.
Bye-bye.